Hello, welcome to Study Hub Pro. All right, let's talk about amino acids. You know that chart, right? The one with all 20 of them staring back at you? It can feel totally overwhelming. I mean, who wants to memorize all that? It feels like this huge, impossible task. But guess what? Today, we're going to crack that code together.
Yeah, it's a classic problem for pretty much every bio or chem student. You've got all these carbons, nitrogens, these weird side chains, and the temptation is to just try and brute force it with flashcards. But let's be real, that's not just tough, it's kind of boring, right? Well, what if I told you there's a much better way?
Exactly. We're throwing out the old school flashcard method. Forget rote memorization. Today, we're going to think like builders. See, instead of seeing 20 totally random structures, you're going to learn how to build them from just a few basic parts. It's way smarter, it's faster, and trust me, this is the stuff that actually sticks in your brain. So you ready? Let's get to it. Okay, first up, the foundation. We're starting with the simplest building blocks. You know, every good building needs a solid foundation, and it's the same deal here. We're going to start with the two absolute simplest amino acids. Once you get these down, you'll see how they become the base for almost everything else. And here it is, our ground floor, glycine. Just look at it. It's so simple, it's beautiful. You've got your standard amino acid backbone, right? The central carbon, the amino group, the carboxyl group. But that side chain, the R group, it's just a single hydrogen atom. That's it. It literally doesn't get any simpler. So remember, glycine is our absolute base. Okay, now this one. This is maybe the most important piece in our whole toolkit. This is alanine. And look closely, what's the only difference from glycine? It's that side chain. Instead of that lone hydrogen, we've got a methyl group, a CH3. That little thing right there is our master key. Seriously, if you remember one thing, remember alanine. It's the base we're going to use to build a whole family of other amino acids.
Which brings us to our blueprint, the alanine family rule. This is our first big memory hack. The whole idea is super cool. We take our master key, alanine, and we just swap out one little piece of its side chain. And by doing that, bam, we can instantly create a bunch of other, more complicated amino acids. It's a seriously powerful system.
Okay, let's see this in action. On the left, you've got alanine with its simple methyl group, right? The CH3. Now, what happens if we grab one of those hydrogens and just swap it out for a hydroxyl group? You know, an OH? Boom! Just like that, you've made serine. See? It's just alanine with an oxygen attached. Super simple.
All right, check this out, because this next move is almost the exact same play. We start with alanine again, but this time, instead of an OH group, we swap in a sulfahydro group, an SH. And if you think about the periodic table, sulfur is right under oxygen, so they're chemical cousins. That one tiny swap gives us cysteine. So basically, if you can build serine, you can build cysteine. It's a 2-for-1 deal.
Okay, let's get a little more ambitious. We're going to take our trusty alanine, but this time we're swapping out a hydrogen for something much bigger, a whole phenyl ring. You know, that big, clunky 6-carbon ring. And what do you get? Phenylalanine. Get it? Phenylalanine? The name literally tells you the recipe. It's awesome.
All right, one last one for the alanine family. We start with alanine. You know the drill. This time, we swap a hydrogen for a carboxyl group. Now, this one's a little different because it's got a negative charge, and that's what makes aspartate an acidic amino acid. It just goes to show how powerful this alanine base is. We've built 4 different amino acids from it already.
Okay, so the alanine family is a huge help, but we're not done yet. Let's expand our toolkit and look at a few more clever memory hacks. These are all about spotting simple patterns.
Okay, this next one is almost cheating. It's so easy. This is valine. Just look at its side chain. See it? Those carbons make a perfect V shape. Valine starts with V. The structure makes a V. You're never going to forget this one. Ever.
So if that's valine, what about leucine? Well, think of leucine as valine's slightly bigger brother. It has the same V shape at the end, but look, there's an extra CH2 group that connects it to the backbone. So I like to think of it as a long V. It's just 1 carbon longer than valine. Simple as that.
Now for a new rule, the +1 carbon rule. Remember how we made aspartate from alanine? Well, we can use aspartate as a new base. If you take that aspartate side chain and just stick one more CH2 group in the middle, you get glutamate. See? It's the exact same acidic end piece, it's just on a slightly longer leash.
And this is where it gets really cool, because this rule has a sequel. Look at asparagine and glutamine. They're the amide versions of the last two. Instead of that charged acid group, they have a neutral nitrogen group. But the +1 carbon trick works here too. Asparagine has one CH2, add another one, you get glutamine. It's the same pattern, just repeated. It's beautiful, really. Okay, let's circle back to one of the big ones we built earlier, phenylalanine. Remember that giant phenyl ring? Well, what if you take that exact same structure and you just add a little hydroxyl group, an OH, onto the far side of the ring? You've just made tyrosine. That's all it is. Tyrosine is just phenylalanine with an OH group tacked on.
All right, so there are a few oddballs that don't quite fit our nice, neat rules. These are the unique ones you just gotta know. First up, proline. It's the weird one. See how the side chain actually loops back and bites its own tail? That makes a really rigid ring. Then there's methionine. It has sulfur like cysteine, but here the sulfur is buried inside a long, floppy chain. And finally the beast, tryptophan. It's the biggest one, and you can't miss that huge double ring structure.
So we did it. We've built all these structures. But you might be asking, okay, so what? Why did we just do all that? Why do these little shapes matter so much? Well, because these aren't just drawings in a textbook. They are literally the building blocks of every single protein in your body.
Think about it like this. The sequence of amino acids is just the beginning. It's like spelling out a word. But the magic happens when that long chain starts to fold up. And how it folds is entirely dictated by those side chains we just learned. Their size, their charge, whether they like or hate water, all those properties force the chain to twist and bend into a specific 3D shape. And that shape is what makes it a working machine, something that can actually do a job in your body.
And this is the take-home message, right here. The final 3D shape, it's all held together by those side chains interacting with each other. The greasy ones that hate water, they all hide in the middle. The ones with positive and negative charges, they snap together like little magnets. Those cysteines can form super strong bonds. And all of these little forces, all based on the structures we just memorized, work together to create a perfectly functioning protein.
So that leaves us with one last big question. Now that you know the blueprint, now that you're a builder, what happens when just one of those building blocks is wrong? Think about a disease like sickle cell anemia. That's caused by swapping just one amino acid for another. The result can be devastating, and it all comes down to one tiny piece being out of place. It really shows you just how important and how beautiful this whole system really is.